So hello everybody, I'm happy to be here and to present our recent results on our research. So can you see the screen, please?
Yes, we can see it, but not in presentation mode so far. So let me present Hanna Lísalová, scientist at the Institute of Physics, Czech Academy of Sciences. The presentation will be from functional biointerfaces to modern biosensors. So floor is yours, Hanna.
Yeah, thank you very much. Thank you. So again, I'm Hanna Lísalová. I'm coming from the Institute of Physics of the Czech Academy of Sciences. It's the largest academic institution in the Czech Republic, and we are covering broad range of areas of physics, starting from astrophysics to biophysics. So our laboratory of functional biointerfaces has been established quite recently, and we are pursuing fundamental as well as applied research on biochip development and biosensors. However, we every time start from the fundamental research on the functional coating interaction with biological systems. We are pursuing multidisciplinary research, therefore it's quite challenging sometimes, but it's very exciting. So far, we have developed several laboratories and built up this very unique equipment, so we are ready for any more chemical, biochemical, and surface characterization challenges in our research. First, I would like briefly introduce our research mission because we are focusing on biomolecular interactions, which is in fact it's a basic element of all the living around. So we are studying this interaction. However, we study this interaction in a connection with some substrate material. So typically one of the interacting partner molecule is immobilized on the surface, and then we study the interaction. Therefore, we are interested in understanding how the surface materials influence the biomolecular interaction and opposite, how the interaction influences the material. So this is the matter of our research. However, we much more complicate this research that we place the system into complex media. We are targeting to using real-world complex media like blood plasma, cell lysis, etc. So these are our basic questions which we are asking in our research. So what are the surface effects on the interactions? What are the biological matrices effects? And how to promote the specific functionality of the non-specific one? So these are the key questions we are asking. So we may divide our research into 3 areas. So we are pursuing basic research on biointerfaces. Then, based on these results, we are designing and developing new biomaterials, and we are applying these results into biosensors. We are developing biosensors for different areas. A common element of our research is biofouling. It's actually a huge global issue in development of any analytical methods. And biofouling can be defined as non-specific adhesion of molecules from complex biological media. As I said, it's a global issue starting from nanoscale, like drug delivery system, through bioanalytical techniques, food packaging, up to ship hulls coatings. So it's a huge problem everywhere, and it makes sense to invest time and efforts to solve this issue. If you look at the example of affinity biosensors, for example, those label-free like SPR, typically if one detects the analyte in a buffer solution, in like model solution, then we can monitor the real-time binding curve. However, if we detect the same analyte with the same concentration, which is spiked into complex sample, the response might be different. It might be hugely increased. And therefore, we are developing the coatings which can, with a direct assay, detect the analyte in complex media, getting the same response as this analyte would be spiked into buffer. So this is one of our questions, and which we are solving. So therefore, we are developing different types, anti-fouling polymer brushes and anti-fouling functional coatings for biosensors. We focus in our research on so-called anti-fouling polymer brushes. This is quite unique and quite exciting material because it combines very high density of chains, like which is used in hydrogels, but it's very robust and relatively simple to prepare. It's also the thickness of this layer is around tens of nanometers, so it's compatible with various techniques like SPR, etc. So we don't lose so much sensitivity as we can lose in case of use thicker hydrogel or thicker layers. So therefore, these polymer brushes are a very interesting approach. But in a biosensor, it is much more complicated because we need to combine two parameters, two features into single material. It is functionality like biorecognition, so we need to typically immobilize some biorecognition element. And on the other hand, we want this surface to be fully repellent to non-specific binding. So it's quite challenging to put these two factors into single material, but we are, I think, quite successful in this challenging issue. The key is the chemical structure. The chemical structure of monomers, which are present in the whole architecture of the polymer brush. We are typically using twitter-ionic moieties, which are somehow incorporated into the structure, and we are mostly playing with the carboxybetaine, which is very interesting because it's a molecule which is hydrophilic and also contains carboxylic group, which can be used as a functional group for attachment of biomolecules. Here's example of designed architectures like different twitter-ionic and non-ionic polymer brushes, and also combine random copolymers of those brushes. So we are trying to tune the physical-chemical properties of the brush to get to the target. Here is the design of our newly developed terpolymer brush. It's composed of three monomers, three types of monomers. Each monomer has a certain role in the final architecture, and very important is also the ratio and the surface ratio of the coating. And what is great advantage of this antifouling brush is that it maintains the super antifouling capability even after immobilization of biorecognition elements like antibodies onto coatings even with very high loading density. Therefore, it's very interesting for biosensor. And I will show you then examples in which we use this terpolymer brush. We have recently patented this structure. Very important is also to understand the molecular mechanism of functionalization. So we develop, for instance, a strategy how to, using certain deactivation agents, we can further improve the fouling resistance of the final coating after the attachment of biorecognition elements. Here's just a figure of our chemical apparatus, which we develop in our laboratory. It's quite unique because we can produce about more than 100 samples per day, more than 100 substrate with different dimensions and different material. Here, typically the methods which use for characterization of the developed coating are infrared spectroscopy, SPR, spectroscopic ellipsometry, and also QCM, quartz crystal microbalance method. Now I will show you examples of developed biosensors based on antifouling coatings. So here's the areas which we are touching. So we are touching medical diagnostics, food safety, and also security. We collaborate with industrial partner in each area. Our first task is to develop the portable biosensor for food safety, for detection of foodborne pathogens. So to get into this task, we first defined the pathogens which are of interest for detection for biosensors. And you can see this is really complicated because there are different classes of pathogens, starting from very small molecules to large bacterial cells. So it's very difficult to combine detection scheme within a single biosensor. Typically, what is typically used for bacterial and viral pathogen detection, the methods are really time consuming, are based typically on cultivation, and also combined with PCR. They need to transport of the samples to specialized laboratories, and they are not much cost effective. Compared to this, the antifouling biosensors can be rapid, can be portable, and can be low cost and even multi-channel. If we summarize the requirements for develop biosensor, we would like to get rapid detection, high sensitivity and selectivity, detection of multiple pathogens in parallel, minimum sample pre-treatment, very robust method, and field use. That's pretty easy, one would say, but it's not. To our best knowledge, there is no commercial device that fulfills all of these requirements together. Therefore, it makes sense to invest time to develop such device. Here are some preliminary, some data which we from our experience. So this is detection of tetrodotoxin, which we did in the past in collaboration with FDA and University of Washington in Seattle. We developed inhibition assay for detection of tetrodotoxin and monitored it by SPR technique. We also developed by SPR biosensor for detection of bacterial pathogens in food samples. We used three-step assay for amplification. We used secondary antibody step and then binding of functionalized gold nanoparticles. And as a substrate, we use like carboxy-beta-based antifouling zwitterionic substrate, and also another modification of this substrate as a copolymer. And we got very interesting results. We got even in very complex samples, it was only homogenized, only liquidized hamburger and cucumber. We detected E. coli and Salmonella with detection limits which were of clinical relevant range. So this was quite good. However, it's still based on the practical use. It's still somehow complicated. How to simplify detection? That was the question, and this is our recent research where I show you how we can go down with the complexity of detection with using antifouling QCM biosensor approach. In fact, we are developing not only the functional coatings, which are dual functional in case of biosensor, we are developing also microfluidic systems and electronics, as well as software, which we also develop some data analysis approaches utilizing artificial intelligence and etc. So combination of all of these efforts can create robust biosensor. Here are some figures of our developed microfluidics. We developed the flow cell, which somehow maximize the probability of catching the target molecules on the crystal chip surface. Also, we developed some switch system, how to minimize the losses of the sample, the liquid, during the transport to the chip. And here is some first pilot of our portable QCM biosensor. Here, how it works. There's two approaches in principle. One approach is that we develop one disposable biochip, which will be dedicated for one pathogen. Or we can develop a cartridge that combines detection of multiple pathogens. Both approaches have some advantages and disadvantages, and we are developing both of them for particular applications. The device should work in that way that we place the chip, which is pre-functionalized with the biorecognition element. Then we inject the samples, and then we simply wash the samples with like standard buffer, like phosphate, like PBS. And in fact, in these two steps, in maximum several tens of minutes, we see the results, yes or no, if the pathogen is present or not. Here are the example of the results. So using poly-CPA surface, we developed the NQCM biosensor. We were able to detect Salmonella in a homogenized hamburger with a limit of detection which was in tens of clone-forming units per milliliter. This is also in the clinical relevant range. Even much better, we got with the E. coli detection in homogenized hamburger. The detection time was less than 20 minutes. We are developing the assay and detection scheme for hepatitis A virus, which is also of interest of food safety specialists. And preliminary results show very, very, very high potential of developed approach also for detection of this virus. And actually, the detection of this virus was somehow prerequisite or that we can probably detect also coronavirus. So last year, when the pandemic starting to be explored, so we started working also on AQCM biosensor for detection of coronavirus. Here is a scheme of the biochip. So it contains again the initiator stem on which is formed interfering polymer brush with terpolymer optimized with terpolymer structure, and then biorecognition element, and then we detect coronavirus in a very complex media like swabs from nasal swabs or oral urine swabs, etc. So this is the idea. This is our enemy molecule, so it contains several proteins, four structural proteins and virus RNA. RNA is the typical target of PCR test, and proteins, especially spike proteins, is the most common target for antigen test. If you look in the advantages, the disadvantage of both standard approaches, so PCR test is very sensitive, can detect virus with a high sensitivity. However, it's time consuming. On the other hand, the antigen test can be cheap, can be fast. However, their sensitivity is rather questionable. Therefore, we are trying to develop a method which combines the advantage of both approaches. So to achieve very high sensitivity, but for very rapid detection, very simple use and very robust technique. Therefore, we develop an assay, which is schematically shown here, in which we detect the N-nucleocapsid protein of the virus. The advantage and the trick, there is a combination of actually two tricks. One trick is that we use super antifolding coating, so there is no non-specific binding. You can see it here on the right figures, that if we injected negative control, what was a very complex cell lysate containing biological species, there was nothing, there was zero response. If we inject positive controls, I mean negative control sample spiked with coronavirus, then we see a apparent decrease in the frequency, what means that there was the sample was positive for coronavirus. And the question is why? And this is the second trick. The second trick is we detect nucleocapsid protein, which is in fact the cells in the virus. It stabilizes virus RNA. So it has certain affinity to RNA. So in fact, we don't detect not only N protein, but also N protein connected bound with RNA. And this is the trick. And we have very tailored antibody, which was developed in our partner institution. And here are the results. So this is example of sensor calibration curve, where we spike the negative control with viruses with coronavirus different concentrations. Here's the resulting calibration curve, and the limit of detection was very nice. It was in the clinical relevant range based on the recent studies, and also it's compatible with asymptomatic patients. Detection time is 10 to 25 minutes, depending what time we need to spend on the washing. We follow up with the testing of real clinical samples. These were nasal swab samples, and we tested 8 samples. 4 were negative according to PCR, 4 were positive according to PCR, and we found 100% sensitivity compared to AQCM compared to PCR. So this is just a very high potential of practical use of this developed approach. And moreover, based on the calibration curve, we can quantify the results. We can somehow quantify the virus concentration in the sample, what can be very, very useful for if we want, for instance, to assess the degree on infection, etc. So this is another feature of developed sensor technology. Here's that we are not limited by the biological sample. Here's example that we continue with example of one case in which one patient, which we measured nasal swab as well as oral rinse, and you can so this is as it is super antifolding. This is the composition of the matrix in which we detect the coronavirus really doesn't matter. So this is the example. It was also a very nice example in which PCR, we were not able to evaluate the sample by PCR because we were told that the donor was a smoker, what somehow complicated the isolation of RNA using standard protocols. So, but for AQCM, it doesn't matter whether the donor was smoker or not. Here, and we continue furthermore, so because we are not limited by the type of the sample, we were requested by Prague Public Transportation State Company to check the safety of the public transportation from the for the coronavirus presence. So upon this request, we performed large-scale study in which we collected about more than 500 samples. We tested first, each sample we tested by AQCM. The majority of the samples were swabs from surfaces which are exposed in public transportation, like in trams, buses, metros, to travelers. Here you can see the figures, the image from the sample collection in which our students were collected the samples. And after the measurement in duplicate by AQCM, each sample was also tested by standard PCR method. I would like to show you the results, but unfortunately I can't because the result is still under NDA, but the results should be available next week. So if somebody is interested, you can contact me, and I will be able to send the results next week. So
I'm sorry, Hannah, I also have to ask you to finish in 1 or 2 minutes because the following presenter, Leo, also needs to leave.
Okay.
So I'm sorry for that.
Okay, no worries. So I will skip some. So just to show, I hope that I showed you potential of our efforts, of our research efforts in development of biosensors. We are now, for instance, collaborating with Czech company, and we are working on development of robotic system for detection of coronavirus. And so next step is a certification process, which is something quite challenging for us because we are new in this, but so we are open for potential partners in this area. So I hope that I show you that based on really basic research on functional interfaces, we can get really interesting biosensor for analysis of real-world complex media. I would like to thank my team. I would like to thank many other colleagues, especially colleagues from University of South Bohemia and Biological Center in České Budějovice, because this the COVID work was done with collaboration with them and many others. I would like also to thank you for your attention, and if you have any question, do not hesitate to send me email. I will be happy to answer. Thank you.
I'm sorry again for the interruption. Is there an immediate short question, short answer? I would maybe have just one very short question. Due to this anti-fouling polymer or brush, the sensor is not disposable, but you can use it for more measurements. So how many samples you can measure with such QCM?
Yeah, this is a very good question. Actually, we are working on development of methods how to regenerate the surface after the positive catch of something. However, we are now working on the method with, especially for the commercial biosensor for COVID, for the robotic system, in which we plan that in case it's a negative, so there is nothing was catch, the surface can be still used. We estimate the number of repeats about 100. So this definitely makes this system much more cost-effective and competent to other techniques like antigen tests. So this is how we now working on.
Okay, thank you very much. Thank you for your presentation. The next presenter is my colleague Leo Schranshofer, scientist at Profactor, with the presentation on polymer-based receptors for biosensors. So Leo, I hope you can make it in the remaining time for you, which is for you.
I try to speed up as much as possible to save some time. So I hope everybody can hear me. Hi, everybody. As Pavel said, my topic is on polymer-based receptors for biosensors, so basically a molecularly imprinted polymers story. Some of you might be familiar with, some of you not. So I will need here some time to explain the concept. Also regarding QCM, I have also something to show a little, so let's stay tuned. Before that, let me first introduce, sorry, some technical issues. Let me first introduce in a very brief manner Profactor. We are a research company located in Steyr and Vienna, about 70 people currently with different research focuses. The one I am from is functional surfaces and nanostructures. So we are dealing on the additive side with inkjet printing, nanostructuring, nanoimprint lithography, but also combining this with the other teams dealing with robotics and robotic assistive systems. So the main topic is molecularly imprinting of polymers. And for those of you who are not familiar with the concept, molecularly imprinted polymers, or in short, MIPs, are artificial receptors based on the key lock principle. So what you do is, in a self-assembly manner, to align functional monomers, the monomer self-assembly around a template. Can be a bacterium, can be alcohol, can be something small, can be something big. Polymerize it and remove the template afterwards. With this, you have a cavity that not just resembles the geometry of the target analyte, but also the complementary chemical information. So you have kind of a negative of, or the chemical negative of your target analyte. There are, of course, some certain requirements in order to do so. So, for example, the most important thing is a high cross-linking degree in order to have a rigid polymer matrix where these cavities are deformed of. There are, of course, some pros and cons to that. That gives the motivation for us to deal with molecularly imprinting polymers. On the one hand, what we identify and what's the strength of MIPS is that the fabrication process is quite straightforward. So it relies just on polymerization. So it's a time and cost efficient way to produce receptor sites or recognition sites. They're relatively easy to implement in sensor systems. Also regarding application, they are highly robust compared to their biological counterparts. So when you think of an antibody, the stability is limited basically to physiological conditions. While polymer-based, you have a much higher degree of freedom, or higher freedom where they can operate. And depending on the applications, you have also high selectivity, which is already demonstrated in literature. However, despite all these benefits, there are also some cons here. There is a catch on that. The one is that due to the self-assembly process, you have a relatively low batch-to-batch reproducibility, and the polymerization time is relatively long when you think of industrial applications. And many of the applications are still on laboratory scale, so more on the R&D level. This gives us the motivation to think of how to industrialize this concept. There is already one known postulated, or not postulated, patented, but also brought already to industrial application by Pilecki and Hauptgroup. But this, if you're interested in, I can explain more, but I think we don't have the time. But we came up with other solutions here. But let me give the main strategy behind first. you can separate this, how to practically generate MIPS in two ways. The first can be referred to bulk imprint, one-step imprint. There are several names in literature, but what you actually do in bulk imprinting is mix everything together, let it polymerize and extract afterwards. So that's really straightforward. So basically what you do is introducing the analyte into a monomeric solution, let it polymerize by itself and extract it afterwards. More controllable is a surface imprint where you just use a template that's immobilized on a stamp, imprint into it and cross-link or cure it during the process, so you have exclusively the binding sites on the surface. When you do what depends on the size of the analyte, so the limiting factor here is basically the diffusion coefficient of the analyte itself. So you need to get the analyte at a certain point out and in again. So the larger the size, the more you go for surface imprinting. There is the particle MIP approach, as said by Pilecki, respectively Haupt. And there is the second one based on a surface imprint I will explain throughout the remaining time, which we call Proto-Mip. So what's this Proto-Mip about? As said in the beginning, you have a self-assembly process, which is basically the bottleneck of the whole thing. You need to, in order to enhance the reproducibility, you need to separate this this self-assembly process from basically from the curing process itself. So what we do here is, on the one hand, coat a sensor surface, can be a QCM, can be whatever layer, plastic layer, glass layer, an SPR substrate layer, with a UV resin that acts kind of the base, and to prepare a stamp with immobilized template and coat it with what we call an anchor group. This anchor group fulfills the role to introduce the functional groups that can self-assembly around the template and can be incorporated into the resin. On merging and curing, and initiate therefore the curing, the anchor groups is immobilized in the resin. The resin is cured and hardened, so it's trapped inside, and releasing the stamp, the cavity is left behind with the functional groups there. Benefit of it is that we can shorten fabrication time. The stamp can be reused by introducing the anchor group again and again after each cycle, and therefore improving the reproducibility and making it industrial relevant for fabrication. So what are the challenges here? I mean, there's a concept, but how is the concept so far in, let's say, development progress? I will give some insight on the individual challenges. Let me first start with the stamp itself. So the first idea was how to show this Proto-Mip, and the best is to use a relatively large analyte that's well established. So we go for bacteria, immobilize them, and try to get a homogeneous stamp, which is what we found out not that straightforward. So we decided to go for E. coli, respectively Bacillus cereus, which are gram-positive, respectively gram-negative analyte bacteria, and use them for stamp fabrication. So we played out a lot with a lot of different conditions, and we ended up with this example where we achieved indeed kind of a monolayer of bacteria on a certain substrate that can be used for stamp fabrication. Here are some conditions. So with a density of about 30%, meaning that 30% of the area is coated with bacteria, which is relatively good and dense for such imprinting technique. The second challenge is indeed to introduce the anchor group. So in order to identify, there are two things basically. On the one hand, to identify the suitable anchor group, and that it's also linkable to a UV resin. So the natural thinking of that is to use a monomer for that can be polymerized in the same way as the UV resin, so basically an acrylate. So with this, we played out with different monomers here, also mixture of that, adding photoinitiators to pre-polymerize to not just have monomers but oligomers, played a lot with and selected them by the potential linkage, respectively potential affinity to the bacteria surface. So therefore we went for, let's say, more hydrophilic approaches than hydrophobic molecules here. Another challenge which gives us currently quite a headache is to introduce the UV resin on basically gold surfaces, respectively metal surfaces, not by, let's say, the adhesion properties or so, and the printability itself. So we want to go for inkjet printing on this. Because this is our main, one of our main research pillars here. But actually to go relatively thin. So the target here is, especially when you go for mass sensitive detection things, is that the layer thickness, so basically the layer on top, basically the volume of the deposit, has an influence on damping effects and on your sensitivity. So the goal was here to go below 500 nanometer layer thickness, which is quite a challenge for inkjet printing, where you usually have layer heights of, let's say, some microns. And it needs to be compatible with the anchor group again. So what was the integration? Here, just a story out of the research project where this is part of. It's a national funded project called Aquanos. This is a schematic of, or preliminary schematic of this microfluidic device where you have a lot of fluidics, you have some electronics, and the black part in the middle, which is called, which has the labeling 8, is the sensor, the sensor itself. Which contains up to 64 individual measuring areas. So it's a multiplexing platform. Of course, you need to do chemometry, but this is a mass sensitive device. So basically, what you do is measuring the frequency, like on a QCM, and it's not selective, but it's sensitive, and therefore you need to introduce a receptor in the end, and this is basically then the MIP. So in a lithographic method, this is deposited onto this chip. So we played out with different things. The main target again was inkjet printing. We thought of two different sensors here. On the one hand, we have our own QCM setup, so we tested our MIPS on our QCM. Here an example of our transducer here, and on the other hand, on the sensor chip by the respective partner in the project, which you see here in a blue colorish scheme, the sensor pixels themselves, so the 64. We managed to coat each pixel individually with inks and get down below 600 nanometer on layer thickness, and also played out with different materials here, what suits best here. Last but not least, the imprinting itself. So this is the key core of the whole thing. There you introduce, or there you craft basically the desired cavities, the desired receptors. So on stamping that as a lithographic method. This is of course depending on the pressure you apply, how to align with the stamp, what's the curing time, the base resin and the anchor group. There needs to be a transfer and so on and so on. So quite a challenge. Our first approach was quite naturally just clamping the things together, but you don't have an alignment control, you don't have a pressure control. The UV illumination, since we are focusing on UV, curing is relatively high and relatively straightforward because everything is transparent to UV what we used. So this is straightforward. This was the benefit. However, as the alignment and pressure control is critical here, we used something what we developed also in-house, what we call stamping tool, where you basically apply via air pressure the stamp onto the dedicated surface. So here we have a better control as well, also with the UV light. Sorry for the background noise if this was heard. We reused the stamp several times since this was a goal. Or basically, let me get to the result here. We reused the stamp at least 16 times with stamping, reintroducing anchor group, stamp again, and so on and so on, at least said 16 times. And here are the two AFM pictures showing that the cavities that are formed are indeed there and are comparable. So also from thickness and so on, from the profiles, you see that they are quite comparable. The average MIP depth is still the same. However, here was a so-called anti-sticking layer used, and therefore we more or less blocked the chemical transfer, the chemical information transfer, unfortunately, which we found out quite later. Nevertheless, we adapted, we optimized to not using such an anti-sticking layer, but tuning with the anchor groups and so on, and with the process itself. Also updated our own QCM measurement setup, which is quite tiny and using two QCMs, one for measurement and the other as reference, containing just a non-imprinted polymer as better reference. And in that way, what we achieved is one of some examples of such a measurement curve. We saw before also QCM measurement, but let me guide you through it. What you see here, I hope you can see my mouse, is that you start with water as matrix solution, and here you introduce through these pipes. In this case, this was done by University of Vienna, which is a partner in this project. On their setup, which is quite not comparable and comparable at the same time, you introduce the Bacillus cereus solution and you have a frequency shift. This frequency shift is proportional to the mass loading on the QCM, which gives then a frequency drop, which is good. So the higher the frequency drop is, the lower the frequency is, therefore you get a better signal. The good thing is that it's reproducible, meaning it's reversible, meaning that you can wash out the bacteria again, introduce again, wash out again, and introduce it again. And on our own setup, the same was done, and this gives comparable results. So here we have around, let's say, 150 to 200 hertz in frequency shift, which we also achieved in our own setup. So this is quite comparable using the same amount of bacteria. So with this, I want to conclude what you heard. Sorry, we dealt with the material and material selection for the substrate. We dealt with stamp fabrication, with the imprinting procedure, on the Proto-Mip approach on generating such polymer-based artificially generated receptors. There is still a long way to go. So we need still to tune the anchor group. in order for bacterial release. So what the drawback of not using the anti-sticking layer is that we rip off some bacteria out of the stamp. So this needs to be still improved. Also regarding introducing the anchor group for, let's say, industrial application, we still need to focus on how to apply them in an industrial manner. So our approach is indeed go for inkjet printing. This would also allow for several analytes at the same time in order to make use of multiplexing strategies and the imprinting process itself, so that we improve the curing time in order to have an improved throughput of the whole thing. With this, I want to thank you for your attention, and of course also want to acknowledge my colleagues who were supporting this development here. Thanks.
Thank you, Leo, also for your presentation. Are there any immediate questions? I don't see any hand raised. So thank you again, and let's continue to the last presentation with the title Diagnostic Processes Through Advancement in Biotechnological Intelligence. The presentation will be given by Daniel Georgiev, CEO at XenoCell. So Daniel, the floor is yours.
So thank you for the introduction, Pavel. Hopefully my camera and microphone are working. Just please confirm.
Works very well.
Okay, great. So yeah, some really exciting technologies out there, many different ways that we're gonna really increase the throughput in cell and molecular detection. And I think Xeno is working along these same lines, but maybe bringing a slightly different perspective to this problem. Let me share so that I don't forget. Share my screen. Okay, so you should be seeing the sort of the cluttered screen, and I'm going to switch to presenter mode. And you should be seeing it in presenter mode. Again, please confirm.
Yeah, we can see it well.
All right, perfect. So a little bit about Xeno. We were founded in 2016, and I would say that we're really a true biotech startup. So I'm also an associate professor at the University of West Bohemia, where I've been running a synthetic biology lab for the past 10 years. And Xeno is essentially a spin-out, as much as the Czech system allows, for a spin-off, I should say, a spin-off from this laboratory, focused on addressing this problem of detecting particularly cellular objects, but applying synthetic biology to be able to detect these objects using living technologies. And I'll say a little bit more about that throughout the presentation. First, a little bit motivation. So basically, the driving factor here is that personalized medicine today is almost synonymous with genetics, really. But if you think about it, paradoxically, your DNA is the part of you that you have the least control over. You inherit the DNA, but the part that is really up to you, how you live, the part that you can change, it's the product of your surroundings. So these are your cells. But personalized medicine today, so in clinical practice, the tools are really missing that would allow us to look at cells. And Xeno wants to change this. So Xeno aims to make cells a computable entity, and by doing that, enable tremendous improvements in preventive health, but also in everyone's lifestyle decisions. So I would say that we're why now? So I'd say pretty much right now we're beginning what will be a single cell decade. The Human Genome Project it's now being followed by the Human Cell Atlas. This is a consortium having over 1,000 researchers across 500 institutions and 50 countries, all working on mapping the cells that make up our bodies. But here I would say that just as the most valuable outcome of the Human Genome Project was not the reference genome sequence, but rather it was the sequencing innovations, NGS, that have since then lowered the cost a millionfold. And similarly, the most valuable, I would claim, the most valuable outcome of the Human Cell Atlas will be a new generation of single cell technologies. To put this a little bit into perspective, modern single cell technologies like flow cytometry, drop-seq, and others are really pushing the limits of what can be measured within a single cell. RNA, DNA, proteins, all from the same cell, but all these technologies are limited by the brute force dogma. So that basically, and then because of this, basically this approach, if you look at the number of samples, the sort of the throughput, this approach is plateaued. And with what we want to do is by bringing some of these real innovations, we want to unleash this sort of single cell data in clinical practice by drastically improving the sensitivity and cost effectiveness. Putting some numbers to these claims. So if you look at the Human Cell Atlas, for instance, to date, approximately 100 million cells costing roughly $100 million have been measured. And with our in-house platform that we have at Xeno, by the end of the year, we're looking to be able to measure or screen through, I should say, approximately 100 times as many cells, but at 0.01% of the cost. And this is really the sort of change that you need to be able to deploy single cell biomarkers and analysis in clinical practice. So the brute force dogma, basically what it means is that in order to today, it's almost taken for granted that in order to analyze one cell, you must first analyze all cells. And now I have sort of a few quick examples. I know we're short on time, but I have few quick examples of technologies that basically abide by this dogma. So here's an example of a hematology analyzer, and this is basically the today's standard for clinical practice. Again, some numbers in the U.S. There's over 500 million blood draws that are made each year. So counting the number of cells, that's 50 peta cells that are sampled each year. And only a small fractions of these are actually analyzed, and they're analyzed by hematology analyzers. It works very simply, sort of the cells are passed through that narrow aperture and they're counted. And in the end, you get counts of the number of monocytes, the number of granulocytes, lymphocytes. So essentially, you reduce the richness of all the single cell data to basically these three abundant populations. But of course, that's just the beginning. Flow cytometry provides much more detailed analysis. And really, flow cytometry is the gold standard for single cell analysis. It's used only in select cases in clinical practice. The general principle, of course, dates way back to the 1950s, and really in its core, and some people might cringe at this, but really in its core, it remains the same to this day. While the technologies have gotten incredibly sophisticated, it does hold true that the more sophistication you introduce with this approach, the more tuning and expertise are required to actually use the instruments. And ultimately, flow cytometer is a statistical instrument with roughly 0.1% to 0.01% sensitivity. So that means you can detect approximately 1 cell per microliter of sample. So that takes us to the absolute state of the art in single cell analysis, which is scRNA-seq, mainly now being lumped together with drop-seq, which was the 2019 Nature method of the year, and it's really based on many fairly recent developments and breakthroughs in microfluidics and nucleic acid analysis. So here's just a quick illustration of how that works. So the cells are encapsulated in these droplets. They're individually barcoded. The droplets are then demulsified and and all the barcodes from these cells are then sequenced by NGS. The real catch here is the cost. In the end, when you sum up the sequencing costs and the reagent costs, it really does come out to be about $1 per 1 cell of data. So extrapolating this to a milliliter of blood, you're basically looking at $10 million per a milliliter of blood. So this is nowhere near a near clinical deployment. Really, sort of summing all of this up, in order for single cells to become a reality in the clinic, we need to be about 4 orders of magnitude more sensitive and about 4 orders of magnitude more cost effective. And this is really what we're after. This is sort of the paradigm change that we're looking to introduce using this living technologies approach. So what makes our solution special? So what makes our approach unique? I would say this is simply common sense, really. If we look at the way we analyze molecules, so it's standard practice today to analyze molecules like protein and DNA with other molecules like antibodies and enzymes. And this works really well, targeted, specific, sensitive, so on. But for some reason, not for some reason, but there's a good reason, but when it comes to cells, the standard practice there is to use large instruments that essentially sort through the cells one at a time. So our patented technology, phenoimmunocomputing, or PIC for short, go by various names depending on what you're picking. So it could be a blood pick and so on. This is the first method out there that uses cells to analyze cells. The general approach has 2 parts. The first step is optional. Antigens of interest that mark cells can be designated in a modular way using commercially developed antibodies. This is basically a standard approach in immunology and so on. The second step, the marked antigens are bound by genetically engineered microbes, which is our technology, that are simply mixed into the reaction. And then what happens after that is automatic. No more, no other intervention is needed. The microbes in this step, the microbes proceed to evaluate the immunoprofiles of the bound cells. During this evaluation, one of 2 things ends up happening. So if the immunoprofile of the bound cell does not match the target profile, then nothing happens. But if the immunoprofile of the bound cell does match the target profile, then the microbes are activated, much like in your immune system, and produce a bright luminescent signal that is easily visible to the outside. So I do have a couple technical slides, and I really didn't know what to expect from this conference. So it seems like this is more of a sort of a technical savvy group. So I will go through these. Basically, there are 2 principles that 2 key features, I should say, that make this technology work, both of which are patented. So first, and this is important, the clusters only form if the antigen expression on the target cell surpasses a given threshold. So this is not the way antibodies work. So in immunolabeling, the level of labeling is proportional to the antigen expression. And you see, and the left figure sort of illustrates the difference between cluster formation, so that means the binding of our microbes to the target cell, the difference between that, which is indicated in blue, and antigen labeling. And so really what you see is that this technology, it works like a gate for antigen expression. And this is important because you simply mix these together and then you let it sit. So there's it's important that all the cells that just have low expression of the antigen are essentially filtered out. And this gating concept is shown It's illustrated again, this time for a particular cell line, HL60, and it's shown and it's shown sort of by this mechanism which of the antigens that are present on the surface of this cell line are actually detected. Those are the ones shown in blue, and which are not. Those are the ones shown in black. And so you can see that there is a residual expression of some of these antigens that are filtered out. Okay, so the second important principle is that following cluster formation, at this phase where the stage where the microbes are activated, triggered, this stage is incredibly precise and incredibly sensitive. So in this graph, in the graph on the left, we've taken a blood sample from healthy donors, and we spiked it with leukemia cells. In particular, here it's the KG1A cell line, and these leukemia cells, the cell line was diluted so that basically in each reaction, in the reaction you either get 0 or 1 of these leukemia cells in the particular reaction, and what this graph is showing that even a single cell in a complex matrix is detectable with high sensitivity and specificity, which is what's shown in the figure on the left. And one of the things that sort of maybe wasn't apparent first, and what makes this, of course, this technology truly a single cell technology, is that it considers the multi-dimensional, multivariate profile that really distinguishes one cell from another. And that's really what really makes this technology generally powerful, is that it is completely modular. So you simply, by mixing and matching the microbes, the user, and antibodies if they're used, the user dictates the target profile that they're after. And the target profile here is represented by a logic gate. So for instance, the gate on the right, which is a 2-input AND gate, this type of a target profile would imply that both of these antigens, both A and B, must be present to trigger the microbes and to generate a positive readout. And the data that just gives you the sort of the fold difference between positive and negative is shown next to the respective gates. And we've used this method to detect various cells in complex samples. These include cancer cells, skin cells, stem cells, blood cells. Really, all cell sizes and cell types can be targeted in this way. And our first product will provide this modular capability to the research community. And this is coming fairly soon, first quarter of 2022. And this figure, I really like it because it's sort of just a historical perspective on how far we've come in the past 5 years. So this started out, like I mentioned, as a basic laboratory principle in 2016. At this stage, we used functionalized agarose beads instead of cell targets, and so you can see the sort of size of those beads that we started off with. And this is how we developed the computational logic mechanisms and the single cell sensitivity. And the figure on the far right sort of shows where we are today. We're able to detect a single leukemia cell, which basically is the size of the smallest human cell outside of red blood cells, in a complex blood sample, basically with sensitivity that cannot be matched by even much more expensive methods. But I would say this is only part of the added value of this approach. The other half, the reason why this technology will make cells the primary unit of information used in medical practice, is that because it will make the retrieval of single cell data cost effective. That's sort of what I mentioned in the beginning. And this is really the other part of the breakthrough is just the structure of this technology itself. So PIC, phenoimmunocomputing, it's the first technology the first information technology that considers multidimensional cellular profiles that is grown. And more, of course, more than that, it is grown using the most widely used industrial organism, baker's yeast. And so we have pilot designs already in place, and it's all leading up to that initial product launch. As I mentioned, the first target is to release this for research use only, but beyond that, we're also working on developing reliable clinical workflows. And we're doing this in collaboration with the Charles University Hospital here in Pilsen. And really the goal here is to enable PIC to become a standard hematology test. So as I mentioned today, you basically get a white blood cell count at best, but using this technology, we're hoping to develop strategic partnerships with hematology test manufacturers to really enable them to, with the same cost effectivity and no sacrifice in throughput, we hope to enable them to basically launch single cell biomarkers into screening pipelines and regular checkup pipelines. And we've received a significant government grant that's currently funding this clinical research, and that will ultimately lead to the certification and market approval of this technology. Okay, so hopefully that was short and sweet. In summary, brute force dogma has been incredible at bringing the knowledge about single cells data to where it is today, but it will not. It's not the right way to move forward. And this approach so the platform that we're developing provides a much more effective access to single cell data that will ultimately enable the use of this data in clinical practice. And then this is the very last slide. So developing these solutions cannot be done by innovations in any one discipline. So we have a very heterogeneous team with expertise in biology, immunology, biochemistry, informatics, mathematics, just many different disciplines that have to come together. And so it's really happy to be working on this and grateful for this team effort. And with that, I open up if there's any energy left, then questions are welcome. If there is no energy, then of course my email I think is going to be in the program, and you can shoot me an email and ask me later.
Thank you very much, Daniel, for your presentation. So is there any energy for questions? Okay, let's leave it to the email conversation for now. Yeah, we are quite behind our schedule. I'm sorry for that, but all the speakers were super interesting. They give really excellent presentations. I'm glad that you stayed until the end of this webinar, and I will for sure inform you about future events which will be organized within NABIAM project. And yeah, thank you for attending this webinar. Thanks to all presenters to their presentation, and have a nice rest of the evening.
Thanks, everyone.
Bye.
Thank you very much. Bye.
Thank you. Bye.
Thanks, everyone. Bye.